turbo engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology. Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to this week's Naked Scientists, the show that brings you the biggest breakthroughs and talks to the major movers and shakers in the worlds of science, technology and medicine. I'm Chris Smith, and coming up, Hantavirus breaks out on a cruise ship. Is this uncharted territory for health officials? Also, why salmon might be hooked on cocaine in our waterways, and NASA's rovers discover a range of new molecules on the Martian surface. The MV Hondius, which left Argentina at the beginning of April, has been dealing with an outbreak of a Hantavirus, specifically the Andes strain of the agent, and several of the ship's passengers have been infected, with fatalities also reported. Others are now self-isolating at home. It's unclear how the outbreak began or whether person-to-person transmission of the virus, which has been claimed in previous outbreaks but is nevertheless extremely rare, is behind at least some of the cases. The World Health Organization has been leading the response, and their chief, Tedros Adhanom Ghebreyesus, had this to say at a press conference on the 7th of May.
8 cases have been reported, including 3 deaths. 5 of the 8 cases have been confirmed as hantavirus, and the other 3 are suspected. Hantaviruses are a group of viruses carried by rodents that can cause severe disease in humans. People are usually infected through contact with infected rodents or their urine, droppings, or saliva. The species of hantavirus involved in this case is the Andes virus, which is found in Latin America and is the only species known to be capable of limited transmission between humans.
WHO Chief Tedros Adhanom Ghebreyesus. Mark Fielder, who's a professor of medical microbiology at Kingston University, has been telling me more about hantavirus infections.
These are an interesting group of viruses that emerge basically out of rats and mice and rodents, and that's where we commonly see them existing, and that's where we get the transmission to humans if we get that transmission from. There is 2 sets of viruses really. There's what we call the old world viruses, which are the ones that exist in Europe and across Asia, and the new world viruses, which are the ones that occur in places like Argentina and Chile and Brazil. And they both present with similar diseases. You get the flu-like illness, which can then develop in different ways depending on whether you've got the old world strain or the new world strain. And the old world strain tends to give you sort of renal problems, gastrointestinal problems, as does the new world strain. But it sort of pretty much ends there. It can be fatal. It can give about 10 to 15% fatality rate, whereas the new world viruses are a little different. They can actually develop into a much more of a hemorrhagic fever and cause more problems. And fatality rate there can be up to 20 to 40%, depending on the patient and the circumstances. So they're quite a diverse group of viruses, but the one we're looking at the moment appears to be a strain called the Andes strain of hantavirus, and that's one of the new world viruses, unfortunately, and it looks like it might have been picked up in and around the area of Argentina.
How does it spread normally? If it's in these small animals like rats and mice and so on, small rodents, how do people get it?
Urine or feces from rodents, and that might be direct contact, or it might be in areas, perhaps in barns or sheds, something like that, where rats or mice have been and have urinated and defecated, and people are sweeping up, and the dust that's formed carries the virus. So that's one way it can get into you. It could also, in rare cases, get into a patient via a bite from a rodent. There have been cases where salivary transfer have got into a patient, but that's rare because rodent bites are a lot rarer. In some cases, we have had human-to-human contact and transmission. When it occurs, it is a rare event, but when it occurs, it's because patients have had very close and possibly intimate contact. So we're talking about people sharing a very close space together, perhaps kissing, lying close together and breathing so that one inhales the other's breath, so as to speak. So there's a number of different ways, but principally for the rats and the mice, it's via the urine and the feces.
How does it actually cause disease? So once it goes in, you're saying it's usually breathed in. That's going to be a respiratory infection. So
Yeah.
how does it cause disease and how does it lead to a fatality when that happens?
It gets into the lungs and starts causing damage in the lungs, which cause the lungs to be leaky, and fluid can enter the lungs and cause effectively something like a pneumonia. And that damage and fluid in the lungs starts to limit your ability to breathe. It can also cause gastrointestinal distress, so patients get an upset stomach and diarrhea. And with all the immune response that's going on, you get myalgia, muscle aches and pains, and you get great fatigue, great tiredness. So all these things brought together is not a great endpoint as far as the disease is concerned, or for you getting the disease. And of course, damage to your lungs is something the body takes great lengths to try and limit and stop and prevent, because your lungs are vital to your survival. So it's that damage to your lungs and ultimately, once it's got through your body down to your kidneys, that can cause a real problem.
What are the possibilities then to explain how these people on this boat got this in the first place?
Yeah, this is a really interesting question, and it is the question of the day, quite frankly, because that's one of the things we don't yet fully know. We know that some of the patients have been touring around parts of Argentina. We know that several people on the boats have been to rural areas as part of their tour. So there's possibilities there that patients could have picked up some virus, perhaps come in contact with urine and feces from the rodents there. And there's been some suggestion that one of the couples had been looking at birds and doing some bird watching, and they went to a landfill site to see these particular birds that they were looking for. And of course, we all know that rodents are very frequent around landfill sites, so there may have been contact there. But at the moment, that's speculation because we don't know. There's been some suggestion that it might have been rodents on the boat. The boat operators have said at the moment they don't recognise that there are any rodents on the boat. I would probably favour this is something that the patients have been environmentally exposed to, so they're on land somewhere and taking it onto the boat rather than the other way around. But at the moment, until a full investigation's done and the full epidemiological studies have been done, we're not entirely sure.
How then will they disentangle a source that was ashore or on the boat from some cases arising because someone else on the boat who's already got it, perhaps they got it from the shore or the boat, gave it to another person? Because all of these things could be happening.
Absolutely. And this is one of the things the epidemiological studies will start to untangle or try to untangle. They'll be interviewing the patients and people on board that they can to try and understand where they've been, what's their footfall before they got onto the boat, and then who have they interacted with, if they can remember, and if that is feasible whilst they've been on the boat. So have a couple or a group of people been walking around a waste site looking for particular birds, or have they not? And if they've not, and they only remained in very urban areas where perhaps the chances of coming across feces and urine from rodents is much less, then it'll start to skew the epidemiological view about where they might have picked things up. And then if they come onto the boat and there's one person with a very low risk background and one with a higher risk background, and they spent a lot of time together, and one couple has got the infection from the other, then they can start to see a pattern and a line where we might see a development of the disease via one particular route of infection rather than another. But at the moment, that epidemiological study will be very much underway with all the patients that they can talk to.
How should they manage this then? Because to my mind, they've got a boat with 150 people on it. They've had a handful of cases of a disease that might or might not be transmitting person to person.
Mm-hmm.
What's the overall risk that's posed? And what should they do with all of these people on the boat who at the moment are perfectly well, but currently not allowed to go anywhere?
I would imagine probably the risk on the boat is moderate. And the reason I say that is because by definition, they can't go away. But that doesn't mean that they are all sitting there cheek to cheek on that boat. They're not, of course, but there is closer contact than there would be if we could send people home. The chances of the virus spreading are a little higher on the boat than they are if they were off the boat, which I think one of the things they'll try and do now is get people disembarked when they possibly can. They'll do an assessment on their general health, and assuming that they are not currently showing symptoms or unwell, then those patients might well be, after a short period of time, sent back home. If they're developing any symptoms, they'll probably be contacted daily and asked what symptoms they're showing, if they're showing any, and as soon as they're showing any minor symptom, they will be taken under medical care and isolated very quickly. But I think that's probably the risk level is moderate on the boat, and once the patients leave the boat, and assuming they're all healthy and all the health checks are done, I think the onward infection risk is very low.
And what, they'd be followed up for the rest of the incubation period, which can be 8 weeks, can't it,
Yeah,
for these viruses? It can be a long time for some of them. So
absolutely.
might be looking at 2 months for someone to sweat worrying that they might succumb to this?
I think that is likely. It's a necessary outcome. We need to make sure that these patients are safe, both from a public health point of view in terms of spreading the disease onwards, and also for their own health.
And the current stance from health authorities internationally is that while further cases might emerge in the coming days, the overall risk remains low, and this is not the start of an epidemic or a pandemic. That was Mark Fielder at Kingston University. To our own evolution now, and US researchers have found that indigenous populations in the Andes carry extra copies of a gene involved in starch digestion. It codes for the enzyme amylase. The adaptation appears to have been shaped by natural selection favouring individuals with extra amylase genes during the transition to living at high altitudes and adopting a potato-rich diet, which happened about 10,000 years ago. The findings have been published in Nature Communications, and Omer Gurkuman at the University of Buffalo has been telling me about his study.
We found that indigenous Andeans have the highest copy number of the amylase gene, and we linked that to a selection that happened maybe 5 to 10,000 years ago, coinciding with domestication of the potatoes.
And amylase, of course, being starch and hence having a gene that helps you break it down would be advantageous.
Of course, that's the main logic, and that link is very exciting because you have a situation where culturally you're changing the diet, and that diet change lead to an adaptive evolution of our very own genes in a population.
Let's look at the origin of all this first then. So if nature and the environment people are living in, which is the high Andes, where they've got, potatoes as their major source of starch, if that's the selection, it must be selecting a trait from a population where that trait already exists. So where did this high copy number of amylase genes come from in the first place, do you think?
The high copy amylase seems to be a specific trait for humans where we have that increase early on in our evolution, perhaps even before we diverged from Neanderthals. But then that variation accumulated. So today, if you look at global populations, we have amylase gene copy numbers ranging from 2 copies all the way to 20 copies. And we are finding that Andeans have almost always much higher copies than other continental populations.
So if I'm reading this correctly then, hundreds of thousands of years ago, which is pre us splitting away from Neanderthals, we begin to evolve more copies of this starch digesting gene. That's in our genome. So the humans that are populating the world have got in their number some individuals with more copies of this gene. And the environments they're finding themselves in might apply pressure so that extra copies sometimes help them. So they end up kind of enriching in certain populations, in certain places.
That's absolutely right.
So if we think then, because the South Americas were one of the last places for anatomically modern humans to get to. So some people get down there in, tens of thousands of years ago, and they take with them this genetic legacy. There's going to be individuals in their number that have more copies of this amylase digesting gene. How does that then get enriched, and why do you think it's 10,000 years ago that suddenly it shoots up in the population? There are more people in the population with that then.
So I think the ancestral population that predates that 10,000 year deadline is they already carry the variation, but you may actually have some people with 2 copies, some people with 10 copies, some people with 20 copies. What happened at that time frame is the ancestors of indigenous Andeans separated from other indigenous Americans, and they end up migrating slowly but steadily, and they start to populate the highlands. And in those highlands, they ingeniously find ways to cultivate potatoes, and that's a very harsh environment, and that becomes a crop, a tuber, that really determines the calorie intake in these populations. So suddenly you have a population that is taking almost all of their calories from potato-based diets. And in those environments, we hypothesize that the individuals that carry much higher amylase copy numbers fare better because they can metabolize starch better, they can control and regulate their sugar metabolism better, and that will create a reproductive advantage that leads to the situation that we observe today.
The archaeology can tell us when the people did the things we know they did and when they began to inhabit these areas, when they began to shift their diet in this way. Is there a similar genetic clock running that enables you to show that this genetic change lines up with the archaeology?
Yes, and that is, I think, one of the most fascinating things, because what we can do with the modern computational tools using whole genome data and contextualizing the amylase locus within that whole genome data, we can estimate when that increase started to happen and what is the trajectory of that increase. And when it started happen, lines up almost perfectly with the domestication of potatoes and the migration of these people into the highlands from their ancestors. And that coincidence is really fascinating.
It's academically very elegant, and it's very satisfying to see that interaction and the fact that the two things have aligned, the two strands of evidence have aligned, and it fits into a coherent story. But what are the other implications of this?
I have to think about this idea of local adaptation as a major force in shaping human diversity, where we have existing variation that we all share, but some of that variation tilts to one way or the other, depending on what kind of environments that we create for our lives. So I'm curious as to other genes and other parts of the genome that behaves similarly, that coincide with other types of environmental or cultural trends. The other thing that is, of course, important is amylase has been associated with multiple biomedically relevant traits, and that means that some populations may be more vulnerable or resistant to certain type of ailments. And these kind of findings will help us to create precision medicine type of leaps in our understanding so that we can have better diagnosis, better treatments, better prognosis for different diseases.
It's a brilliant bit of detective work, isn't it? Omer Gurkuman there at the University of Buffalo.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
This is the Naked Scientists podcast with me, Chris Smith, and still to come, how NASA's rovers are searching for signs of ancient life on Mars. But first, do stay on the line, because we have an environmental story which might have you hooked. Ba-boom. And that is because scientists have found that salmon exposed to cocaine and its breakdown products in polluted rivers show significantly altered behaviour. Marcus Michelangeli at Australia's Griffith University exposed freely swimming test animals to drug levels that were very similar to those that fish are encountering in the wild, and he got quite striking results.
When we take pharmaceuticals or drugs, they break down into essentially components, which we call metabolites. Cocaine primarily breaks down to benzylcogonine, and this is the metabolite that we often find at high concentrations in the environment, along with the parent compound, cocaine. So that's why we focused on this metabolite and the parent compound, cocaine, for this study.
And how did you actually do the study?
We tracked 105 juvenile Atlantic salmon in Lake Vättern, which is Sweden's second largest lake. Some of these salmon were exposed to cocaine. Some of the salmon were exposed to the metabolite. And then we had control fish exposed to a facade implant. And the way we exposed these fish was that we implanted them with a small chemical implant, which essentially releases the contaminant into the fish's muscles. And this means that we could chronically expose the fish and test their behavior over time while tracking them in this lake.
And you know that when you do that, the fish is exposed to a concentration of drug or metabolite equivalent to if it were just swimming in an environment with a relevant concentration in it.
So we actually performed a side experiment as the main experiment was going on to actually test the efficacy of these implants. And we did find that the exposure in this side experiment did meet our aims of meeting an ecologically realistic exposure.
How do you know where the fish go and what they do once you put the implants into them and let them go?
We used a technology called acoustic telemetry to track the fish behavior. So essentially, we set up receivers in Lake Vatten and we implant the fish with a tag which sends a signal to the receivers and it tells the receivers the location of that fish. And this allows you to get incredibly high resolution location data on the fish's movement. So we were able to detect the fish every 15 seconds as it was moving around this lake. And so this can give you really fine detail information about how they move and behave within the lake system.
And where they go as well. So you can follow them over time. Because I suppose that's another powerful aspect of this. You can see not just what they do, but how long they do it for and where they do it.
Exactly. So if you have high quality information about the habitats in the lake, you can also look at how they use different habitats. In this system, we didn't quite have that information, so we couldn't pinpoint the types of habitats they were using. However, we could see how far they were dispersing in the lake. So, for example, we found that the salmon exposed to the metabolite actually showed the most dramatic behavioral effects. They ended up dispersing up to 14 kilometers further in the lake than control fish, and they moved 2 times the distance that control fish did per week.
So they're more active. They seem to go further. Why is that a bad thing?
Any change in an animal's behavior that is not a natural change is not a good thing. Behaviors have evolved in these animals over a long period of time. So, for example, an animal that is moving a lot more than it used to be doing suggests to us that it is allocating energy towards this dramatic changes in movement that it would be otherwise allocating to other important factors in its life history. And so this can have important implications for not only the survival of that individual animal, but also the population that it's part of and the community that it's part of.
What sorts of waters are going to be affected in this way? Is there cocaine everywhere, or are there hotspots? And therefore, are there some areas where we should focus because there's a high likelihood of particular vulnerability in certain species?
With cocaine in particular, we find it all around the world, and it's particularly at high concentrations where you find more people, so in our urban centres. And so the wildlife that are living in these environments, these habitats, are more likely to be exposed to drugs like cocaine and, of course, other pharmaceuticals we also find in the environment.
In recent years, we've seen examples of people looking at antidepressant drugs, which we out, contraceptives, which we out. They all end up in the water. So presumably this is like a canary in the coal mine in some respects. You're showing there is an effect of cocaine. That's one drug, but there are many others that could equally be having an effect.
That's exactly right. So many drugs and pharmaceuticals that we take, they end up in our wastewater. And unfortunately, at this point in time, many wastewater treatment facilities do not have the technology to adequately remove a lot of these pollutants from our wastewater. So they end up getting discharged into our waterways. So another real hotspot for finding these contaminants is downstream from our wastewater treatment facilities, because wastewater treatment facilities are discharging the wastewater into rivers and creeks. And so the closer the water is to a wastewater treatment facility, the higher we find concentrations of illicit drugs and pharmaceuticals.
What do we do about it then?
I suppose there's no silver bullet, but the good news is there are ways of mitigating the impacts or reducing our impacts. Cocaine, I suppose, a little bit more difficult because there is already tight regulation. It's a legal drug. But the main thing that we can do is invest in better wastewater treatment technology. So a lot of wastewater treatment plants in richer countries have better wastewater treatment. So they have a 3-step process for treating water. But of course, not all facilities have this 3-step process. Some only have a 2-step process. And that's where we find these micropollutants being able to escape into the waterways more regularly.
Marcus Michelangeli at Griffith University there, and that study was published in Current Biology. NASA's Curiosity rover landed on Mars in 2012 with the aim of adding to our understanding of whether the red planet has or had in the past the environmental conditions to support the growth of microbes. Meanwhile, the Perseverance rover, which landed in 2021, aimed to look for signs of ancient life. Since then, experiments have discovered a whole range of organic molecules on the planet's surface, although we can't say which molecules were preserved from Martian life and which were formed through geological processes or originated from meteorites. Nevertheless, their discovery confirms that ancient Mars did have the right chemistry to support life. Amy Williams is at the University of Florida, and she's been working on it.
So I am an astrobiologist, and I'm interested in whether there's life beyond Earth. And one of the ways that we can explore this is by searching for the components that make up life as we know it. One of those things is organic carbon. So searching for those on planetary bodies beyond Earth is one of the ways that we can start to explore that question.
And how is that actually occurring?
Well, there's a bunch of different techniques that people use to basically detect these kinds of molecules. But the way that my study has worked is that we actually get to do this work in situ or in place on Mars. I work with the NASA Mars Curiosity rover science team. And that mission actually has an instrument on board it that's able to detect organic carbon. And so I've had the opportunity to work with the team to develop an experiment that actually revealed some organic carbon that we've never seen before on Mars. And so it's a way of sort of expanding the library of molecules that we know are present on Mars.
And what are the molecules?
One of them, I'll give you a big term here, is benzothiophene. So this is a molecule that's made up of rings of carbon, and that molecule actually has a sulfur element within it. And that actually tells you a little bit about where the organic carbon might have come from. We actually think that it came from meteorites that rained down on Mars billions of years ago.
How was it actually formed then? Was it cooked up on the meteor, on the parent asteroid that gave rise to the meteor, or did it form when it hit Mars and it was the conditions created during the collision that created it?
It's hard to say. We think that some of the molecules that we saw, so we saw more than 24 molecules revealed with this first of its kind experiment done with the Curiosity rover. And some of the molecules have the same character that you see when you do this type of experiment with a meteorite. And so we think that these organic molecules formed on the meteors in interstellar space. And when they rained down on Mars, those organic molecules were actually able to be preserved over very long time periods.
Why did we not see them before?
So the experiment that the Curiosity rover performed is a first of its kind. It uses a chemical that can react with sort of our powdered drilled rock sample. And it has a special capability to break apart the components of the rock and break apart actually more complex organic carbon, like big molecules, basically, and break that apart into their little constituent parts.
What does this tell us then in terms of the likely prospects of life processes having existed on Mars? Is this a sort of an opportunity to say, well, we know what can form in space, therefore what's probably present on Mars, and what might not have come from space and therefore might have been made on Mars and might be significant?
The fact that there was this basically more complex organic matter that we broke apart with this first of its kind experiment, that tells us there's more complex carbon preserved on Mars than we realized previously. We just didn't have a technique to see it until we did this experiment. That's really, promising in the search for life beyond Earth, because it means that, we expect life to make complex organic molecules. And if that is preserved in the rocks on Mars, we've sort of demonstrated that it could potentially survive over billions of years, and it could be detectable with the techniques that we have on board this mission as well as future missions to Mars.
So this is a kind of telltale. It says we can do this. We can detect long-lived species regardless of where they've come from, and the technique works. So does that mean that this opens the door to then going after life-linked molecules next then?
The hope is that we would be able to search for more complex molecules that could be indicative of life, or at least linked to life. One of the molecules that we did see with this experiment, we weren't able to exactly identify it, but we have an idea about what it is, and it actually is a precursor to some of the molecules that make up DNA, the genetic code within all life as we know it. So it's not evidence for life, but it tells us that basically the components that we know life needs were present and preserved on Mars in the ancient past.
Now you've proved you can do this. Is it just widening the search, or are you going after a specific target?
So this particular experiment contains a chemical that we had to send with the rover when it launched more than 13 years ago now. And that means that we used one of the two experiments on board the rover for the study that I'm talking about today. But what this does do is it sets the path for us to be able to do this experiment both one more time on Mars with the Curiosity rover, as well as with future missions. So this chemical is actually flying on board the European Space Agency's Rosalind Franklin rover, which is hopefully going to launch in 2028. And it's flying on board the Titan Dragonfly rotocopter mission. So we're looking at, very different planetary environments, but we're using this experiment as sort of the springboard to say, hey, it worked. Here are some lessons learned. And what more can we learn about the chemicals that make up life and whether there's life on other worlds based on using this experiment?
One of the most profound things I learned in the last year or so from talking to researchers analysing bits of the asteroid Bennu, which we're now privileged to have back on Earth because an operation was mounted to go there, get bits of it and retrieve them, is that there was chemistry going on inside these asteroids. There was enough warmth in there to have liquid water and chemical reactions. They weren't just cold, frozen, inert, icy bodies and rocky bodies out there. They were clearly making things, and some of them were really quite complicated molecules that were then raining down on planets like the early Earth and Mars. So it really sort of opens your eyes to what might have been happening early on then in the solar system and where really the building blocks of life could have come from.
Absolutely. I think it's that type of return sample mission that gives us really profound insights into what the solar system was like in its infancy and what the ingredients were that might have led to an origin of life on Earth and potentially on other planetary bodies.
It's arguably one of the biggest questions out there, isn't it? Amy Williams at the University of Florida. Now, in parting, Sir David Attenborough marks a century not out. The legendary science broadcaster and writer, who was born on the 8th of May 1926, has inspired countless people around the world to take up science, including me. And I'm very proud to say I have met and chatted with him a few times, and he was very kind to introduce himself to me, just in case I didn't know who he was. What a gent. We wish him a very happy 100th birthday. That's it for this week. Do tune in on Tuesday when we'll be examining the Hantavirus outbreak on the cruise ship in a bit more detail and finding out how the command centre at the World Health Organization are managing the situation. Before we go, thanks very much to all of you who are supporting us with donations. If you'd like to get behind us and show us your appreciation and you enjoy what we do every week, do please head over to nakedscientist.com/donate. We're very grateful. I'm Chris Smith. Thanks for listening, and until next time, goodbye.